Mana, ingareo, ingata wira pumanawa, vro rakatira ma, tenakoto, tenakoto, tenakoto. Norera, ko waio, ko Harleen Hain, takawingawa, ko te tumuakio te wharewanaka o atago aho. Namihi nui kia koto, norera, tenakoto, tenakoto, tenatato katoa. My name is Harleen Hain, and as many of you know, I have the great privilege of being the Vice Chancellor here at the University of Otago, and it gives me great pleasure to provide some opening comments at this inaugural professorial lecture for Professor Greg Anderson. On behalf of the university, I would like to begin by extending a particularly warm welcome to members of Professor Anderson's family who have joined us this evening for this very special celebration. His wife Vivian is here. His children, Lydia, Lewis, and Finlay, are here. His mum is here, and his father-in-law is here. And a number of these people have traveled quite a long distance in order to share in this celebratory event. I'd also like to acknowledge our Chancellor, Dr. Royden Somerville, who is also in the audience this evening. Against a backdrop of an incredibly busy day job, Royden also does an immense amount of service for us here at the University of Otago, and I think it's particularly fantastic that he makes time to come to these inaugural professorial lectures. So thank you, and thank you for being here, Royden. Now, in addition to all these very special guests, as I look out on the audience this evening, I recognize many of you. There are staff and students from around the university and members of the wider Dunedin community, and it is fantastic, as always, to see so many of you here. No mai, hadamai, welcome. So we've all gathered here, members of Greg's family, members of our university community, and members of the wider Dunedin community, to celebrate Greg's promotion to professor. Now, as many of you know, the path to professor here at the University of Otago is not an easy one. It requires years and years of excellence in teaching, research, and service, not only to the university, but to the wider community as well. So as you will learn this evening, Professor Anderson is an effective and highly motivating teacher, and he is a very popular postgraduate supervisor. He teaches both very large 100-level courses here at the University of Otago and also small 400-level seminars. He's also the past recipient of the Health Sciences Postgraduate Supervisor of the Year Award. Professor Anderson is also an outstanding researcher who has made major contributions to research in the area of brain regulation of fertility, and I'm sure that we will hear quite a bit about that this evening. And last but not least, Professor Anderson has provided sustained service not only to his department, but to the university and at a national level as well. So Greg, on behalf of the University of Otago, it gives me great pleasure to congratulate you on your very well-earned promotion to professor. I will now call on Professor Vernon Ward, who will tell us just a little bit more about Greg's journey to professor. Norera, tenakoto, tenakoto, tenatato katoa.
Thank you, Vice-Chancellor. Tēnā koutou katoa, hami nui tēnā kia koutou. Welcome everybody to Professor Greg Anderson's inaugural professorial lecture. I just want to thank you very much for coming along this evening and celebrating this occasion with Greg and with the rest of us here. And it's a pinnacle of an academic career to make your way to professor, and he's well and truly earned that right. It's my role as the Dean of the School of Biomedical Sciences to provide some little bit of background information on Professor Anderson for you this evening and his career to date. For those of you who aren't aware of his background, Greg was a graduate of the Lincoln University, getting his Bachelor of Agricultural Science in 1993, and then he went on to get his PhD in 1998. And this is something I actually didn't know myself, and it was around neuroendocrine control of reproduction in red deer. So it's good link to here. He undertook a postdoctoral fellowship at the University of West Virginia in the USA, and then he returned to New Zealand to take up a Foundation of Research, Science and Technology postdoctoral fellowship at Massey University in 2000. He came to us here in Otago in 2002, and he was a research fellow before becoming a lecturer in 2007, and then subsequently has progressed through senior lecturer and associate professor to reach professor today. In his 20-year career, from attaining his PhD to becoming a professor, he's really worked his whole time in neuroendocrinology. And it's quite appropriate, and I make the point that he's not only in the Department of Anatomy, he's also in the Centre for Neuroendocrinology, one of our top flight research centres here at the university. He's been extremely successful in gaining research funding. He's had near continuous Marsden funding as a principal investigator since 2003, and there's not many that can say that. He's received as PI HRC project grants. He's been an associate investigator in multiple HRC programs, so PI's project grants, and he's on a number of other research funding as well. His H index is 29, with over 2,800 citations and 56 journal publications. And you can see the strength of those publications as they're sitting there in neuroendocrinology, endocrinology, Journal of Neuroscience, core contributions to his field with a substantive body of work. Consistent with that focus, Professor Anderson is currently president of the New Zealand Endocrinological Society, and he's on the editorial board of the Journal of Endocrinology at present, and has served on a number of other editorial boards in the related field. He's supervised over 30 postgraduate students, and as the Vice-Chancellor pointed out, he was voted the Health Sciences Postgraduate Supervisor of the Year in 2011. But I do want to draw a little bit more, as already been mentioned, on his service to the department and to this university. He teaches widely in anatomy, neuroscience, and into the medical program. He's the Deputy Head of Department of Anatomy, which is an incredibly large and complex department, where he oversees the teaching through that department, and that's a substantive role. He is the Chair of the HTRU Academic Advisory Committee. He's the Chair of the IARMS Process Group. He's the member of the RSF Building Executive Planning Team, and he's served on the University of Otago Animal Ethics Committee, so he's contributed a lot over his time here. When asked to describe his research, what he's got here is his big picture goal. And his big picture goal is the development of new improved treatments for infertility and anxiety-related disorders, both of which are unseen conditions experienced by a large proportion of modern societies. So I hope that big picture fits with what you're going to present. And for this IPL, he's going to explore the neuronal and hormonal mechanisms that coordinate to drive ovulation. He will look at the lack of ovulation due to either too much or too little fat, and the lack of ovulation due to stress. And the idea here is to explore and find workable treatment options for these conditions. With that, I'd like Professor Anderson to present his inaugural professorial lecture, and I just want to thank him for a nice tongue-twisting title to work me over at the end here. Female Fertility, Fundamentally Faithful Yet Frequently Fickle. Got it. So, nō reira, tēnā te mihi kia koutou katoa. Tēnā koutou, tēnā tātou katoa.
Tēnā koutou katoa, ko Greg Anderson, tōku ingoa. Thank you, Professor Haynes and Ward, for your kind words of introduction. Thank you also to my head of department, Professor Matthew Smith, for being here, and also Professor Blakey, Professor Squires and Professor Nicholson, Vice-Chancellor, and to all of you for coming along. Thank you very much. It gives me great pleasure to have both friends and colleagues and family here today. Special thanks to all those who travelled to be here. It makes it special for me. So, for my parents and grandparents, infertility was never an issue. And this is my mother's family. My mother was the youngest of 7 here. She was born deep in the south in the Catlins. And I like to think of my mother as a would-be academic. She was always loved books, ducks of her school. I think there were 2 students in her year at Awaka High. But, you know, university study was never really an option for my mother at that time. And so my mother was a a teacher aide and a nurse aide, and of course the mother of my family. My father was also the youngest, a more moderate-sized family, so this is my father here, the youngest of his brood as well. I never described my father as an academic. He dropped out of high school just as soon as he could. And yet, and so Dad couldn't be here today. Dad's in hospital with advanced Parkinson's. And although, you know, books were never my dad's thing, even when his brain was ravaged by Parkinson's, my dad was the person you would go to for the spelling of a difficult word, what 13 times 45 is, how to solve a crossword puzzle. He was always the fastest of these things. So I think academic ability comes in many shapes and sizes, and Dad would have loved to have been here. He also, you know, he could also play me under the table for table tennis, even when he got to the stage where he couldn't bend down and pick up the ball. So some things are just retained in the brain. This is the family that my mother and father raised, so I'm this one here, if you can't still recognise me. And so you can see that, you know, infertility was still not a problem, even at that stage. And my brother Craig is here today, so welcome. And this is my family, so this is my wife Vivian, my son Finley, my daughter Lydia, and my son Lewis, and they're all here today, come from as far as Auckland and Wellington to be here, so it's great to have you all here for this lecture. Thanks very much. So, 3 generations of Andersons represented in these photos. So, I need to give you just a little bit of background, and I don't want to make this too heavy on science, but you need to know just enough to be able to get you through some of the science that I'm going to present later on. So here's a little bit of background to the control of female fertility. Most people understand fertility to be controlled by the gonads. So in the female, of course, that's the ovaries, and so during the first half of the cycle, there's a clutch, maybe 20, of eggs that begin to grow, and only one of them will make it to this stage where it's a large, or the egg doesn't change that much. This is the egg here, but the little bubble around it's called the follicle, and only one of them will make it to that large stage by mid-cycle. As it grows, it releases estrogens, and they're critical to the control of the events that happen at that time and to follow. And at that time, at mid-cycle, the egg ovulates, and the cells that are left behind, all these cells here that are left behind, form a new structure on the ovary called the corpus luteum. The egg is released into the fallopian tubes here. Perhaps it will meet with a sperm and be fertilized and implant in the uterus. But the structure that's left behind begins to secrete a different hormone called progesterone. And progesterone dominates the last half of the cycle. It's easily measured as evidence, hormonal evidence that ovulation has happened, and I'll show you plenty of examples of cycles where you can see progesterone going up and down, and you can read that as evidence that ovulation has happened. Here's a picture of ovulation happening. I think this is in a rhesus monkey on the cover of a recent endocrine magazine, and you can see the rupture of the ovary here, the egg being released along with all these cells around it. And this is a short video of human ovulation that has just happened, and you'll see the egg here just in the process of being released and being picked up by these little fingers which will guide it into the fallopian tube. So you can see the massive rupture that happens. It's a bit frightening to see this on the ovary, and the egg is released and it's hanging on by a mucus plug, and it's guided into the fallopian tube by these little fingers called fimbriae. Well, while that's all integral to my research, this is not actually where I target my research at. I'm a neuroendocrinologist, and so for me, control of reproduction is all about what happens here, and a small part of the brain called the hypothalamus, and a little gland underneath it called the pituitary gland. Hypothalamus, pituitary gland. And so these are the parts of the brain and the gland under it which direct the events that are happening down here below the belt. And when I say they direct it, you can argue that it starts at either end because it's a cycle. These hormones feed back on the events that happen at the brain. So the pituitary gland here releases a hormone called luteinizing hormone, and these are just some little windows into what happens throughout the cycle. Early on in the cycle, the pulses are not too fast, quite large pulses. Towards mid-cycle, they get faster, smaller but faster, and they eventually culminate in a massive surge release of LH. And this is the central event for a neuroendocrinologist of the cycle. And I always try and tell my students not to call these surges and not to call this pulses because they're fundamentally different. So this is a surge, these are pulses, and perhaps the problem is that the word surge is a bit understated. Someone recently in an article I was reading described this as a luteinizing hormone tsunami. So I'm going to refer to it as a tsunami so that you don't get mixed up between surges and pulses like my students do. And after this tsunami, this is the thing that drives ovulation. You need these massive amounts of luteinizing hormone in order for this egg to be ovulated. And after that, LH pulses return to a much more slow, drawn-out state. Now, most of the time, this whole system is kept relatively controlled and under homeostasis thanks to the feedback effects from these steroids. And they have negative effects on controlling the secretions of luteinizing hormone. And so that works well for homeostasis because every time LH starts to get too high, it drives an increase in the release of these steroids and they dampen things down again, and it keeps it nice and stable. But perhaps you can see that that's not what's happening here. At mid-cycle, the level of these estrogens are getting really high thanks to this large follicle that's releasing massive amounts of estrogen, and yet at the same time, we're building up to this LH tsunami. So somehow the negative feedback has broken down, and there's a remarkable switch to positive feedback where instead of inhibiting these events, this here drives this event. And just how the brain can switch its state in its response to this hormone in just a matter of hours is a remarkable phenomenon that we are still slowly beginning to understand, but I'd say we're a long way off it yet. So for neuroendocrinologists, reproductive neuroendocrinologists, this is really the central wonder of our lives, perhaps, how this event happens. It's easily measured, so you can, if you know what time to sample, you can measure this evidence that ovulation is about to happen. And even in humans, I was in the supermarket the other day, and you can buy little urine tester kits, so metabolites of this hormone, luteinizing hormone, are available, are detectable in the urine, and you can check whether ovulation has happened simply by a single sample. Obviously, you would never get this profile. You get a smiley face if ovulation has happened, and looks like a blank face if it hasn't happened. That's what's happening at the pituitary gland. Higher up here in the hypothalamus, there are a small set of neurons, and they're called gonadotrophin-releasing hormones that are mirroring the effects of these. In fact, they're driving all the effects of these. And so these are neurons, but they secrete their product into the bloodstream, so they're actually making a hormone. That's why they're called gonadotrophin-releasing hormone neurons. And every pulse of this LH will be driven by a pulse of GnRH. The tsunami, will be driven by a tsunami of GnRH. But it's much harder to measure because these are brain neurons and they only secrete a tiny amount of their peptide. It does make its way into the bloodstream, but only a little local bloodstream that links the hypothalamus and pituitary gland. And so it's extremely difficult to measure these things. But in the species that have been done, and that doesn't include humans, it exactly measures these effects of these patterns of luteinizing hormone. All these profiles here are samples that I've taken throughout my career, and none of them actually are from women. In fact, I don't think I've knowingly blood sampled a human being ever before. So all my research is based on animal models, and yet the wider aim, as Professor Ward said, the wider aim is to become more educated about human fertility, how it's controlled, how it goes wrong, so that we can develop better treatments. And I operate well back from there, and perhaps the people other than me will take that information and develop into clinical trials, but nevertheless, the build-up of knowledge based up on these animal models contributes to that wider goal. So it is amazing that such a complex thing that involves temporal timing as well as multiple different layers from the brain down to the ovaries can work so faithfully just about every time. And so women can cycle every month for their entire reproductive life, just like clockwork. Many women will have this suppressed because of the contraceptive pill for much of their reproductive life, and they can stop taking that pill, and the very next cycle they can get pregnant. So that's why, as I think about that, I think it's a remarkably faithful system, and it amazes me that it works so very well. But that's cold comfort, perhaps, for those many couples who struggle with infertility. And it's a surprising amount, up to a quarter of couples in New Zealand, as well as other parts of the world, experience infertility, defined as not being able to conceive after 5 years of trying. It's a silent problem. People don't tend to declare their infertility, and others perhaps just think they don't want to have kids. But it's a distressing state to be in, not to be able to conceive. So it's a big problem. About 50 million couples are unable to have a child after 5 years of trying. And sadly, as someone who works doing research in this area, we've been able to do very little to change these statistics over the last 3 decades. We have IVF, of course, and that's wonderful for the few people to whom it's available, but on a population level, this high level of infertility has stayed the same for decades, and there's been few real breakthroughs. So it's a big problem. Well, I'm going to give you 3 little stories or vignettes that span the things that I've been doing throughout my career. And the first one is about a type of infertility that's really nothing to do with humans, and yet there's things we can learn from that that might have relevance to ourselves. So, as Professor Ward said, I began my research looking at seasonal patterns of reproduction that serve to time the birth of the young to the appropriate season for the survival of a baby, whatever. And it's been called nature's contraceptive by someone other than me. The next story I want to tell you about is the links between nutrition and fertility, which is perhaps an increasing problem in our current generation. And the last one is the links between stress and fertility. Again, a very common problem, and we've been working on establishing what the links are that might bring about infertility because of these states. So let's start with number 1. So this was work that I did as part of my PhD. Actually, before I get to that, this is some background that was done around about the same time to help you understand how seasonal reproduction works. So this is data from New Zealand, so you don't have to think about changing the hemispheres. But in deer, which I did my PhD research on, they become fertile around about April, when days are getting shorter, this time of year now. And you can tell that from these because you can see that there are cycles of progesterone going up and down about every 2 weeks, and this is the length of the cycle of a red deer. So every one of these, as you now know, is evidence that ovulation has occurred, and it's happening in the red deer here 8 times, just like clockwork, unbelievably faithful, and then it stops. As the days start to get longer and spring approaches, it stops. And so this is in the absence of the hind getting pregnant, which is probably an unusual situation either on farms or in the wild. This animal will just continue to cycle, but only throughout the winter. And the same in many other species of deer. So I think the clockwork of this system is a remarkable demonstration of the faithfulness of reproduction, not only that it occurs with such wonderful regularity, but the fact that it starts and stops, and every animal in this experiment had almost exactly the same timing of reproduction. So it's a fascinating thing to study for the faithfulness of the control of reproduction. So I began my PhD research and completed it under the supervision of Dr. Graham Burrell at Lincoln University, a little university just south of Christchurch. And Graham Burrell was one of my undergraduate lecturers, and I always think of him fondly when in my own teaching, because he's an example of a guy that can inspire you in his undergraduate teaching. And it was during those undergraduate years in his classes during my Bachelor of Agricultural Science that I determined that I didn't want to be a farm advisor or anything else that I could have become with that undergraduate degree, but I wanted to study reproductive physiology. And it was because of his passion for his subject. And so if I can impart some of that to my students, I think I'll be doing well. So the background to this story comes before I came along, and Graham was doing some work with a previous student of his who was looking at the role of the parathyroid glands and the thyroid glands embedded in the neck, and the role that they had in, I'm not sure what they were studying, to be honest, but their studies involved removal of the thyroid glands, and the animals remained remarkably healthy, and I guess they got their results. But one thing that they noticed along the way that completely surprised them all was that while the normal animals went through the typical cycles that you may be aware of that deer do of body growth, antler hardening, shedding their antlers, and so on, the animals that had had their thyroid glands removed did not. They also remained remarkably healthy, but once they got into this state where you can see their larger body size, their hard antlers, you can perhaps imagine that this animal's attitude is targeted 100% to mating and the other 100% to fighting off other rivals. They stayed in that state for as long as the experiment went on. And remarkably, they never made the transition to this state. And it was like whatever it is in the brain, presumably, that switched them from their breeding state to their non-breeding state failed to happen. It was a remarkable chance observation. So I began my PhD with Graham Burrell investigating whether thyroid hormones are critical for that kind of transition in female animals. And we had looked at the profile of thyroid hormones. This is T3, the active form of the thyroid hormones. And although it's a bit up and down, you can see that it increases here as the summer months approach, and it peaks just before midsummer. And we wondered whether these increasing levels of thyroid hormones might be the thing that drives the animal to somehow change the neuronal wiring of its brain so that it could enter the non-breeding state, as I showed you in those previous graphs. So our treatment involved removal of the thyroid glands, and again, the animals remain remarkably healthy, and you can see that their thyroid hormone levels were right down at assay detection levels. These are the animals, and the only thing you could notice just from looking at them is that the thyroidectomized animals retained their coarse, pale winter coat. We had another group of animals where we replaced thyroid hormones, so they were also thyroidectomized, and we replaced the thyroid hormones. It's a classic endocrine, old school endocrine experiment where you remove the gland and replace the hormone that you think is the culprit, and see if you can revert the animals back to the control state. So here are the results, and these are profiles of progesterone, just like I showed you before, and every significant episode of progesterone is evidence that the animal has ovulated. So the control animals, just as I've shown you in other data, had these cycles right until spring, and then they stopped, and then progesterone was low throughout the summer months. No surprises there. The big surprising thing was that all of the thyroidectomized animals, bar one, which we have evidence had regrown its thyroid gland, all of them carried on cycling for as long as we could measure for. And this really blew us away, that the animals could not only stay wonderfully healthy, but actually stay in their fertile state for as long as we could measure them for. So whatever causes this transition is not active in the absence of thyroid hormones. And when we replaced the thyroid hormones, we were able to terminate their breeding season at exactly the same time as the control. So clearly, these thyroid hormones are needed for these seasonal transitions. Here's another example of these profiles of progesterone going up and down each cycle. And we were able to show that the animals were able to be mated, and so most animals will only mate just before ovulation, and so we could synchronize ovulation in some of these animals and observe their mating behavior. So I spent long hours of my PhD in this position with binoculars, observing mating behavior in red deer. And these animals were able to be mated even out of season in the summer months. Quite some challenges getting the stag to be interested in them in that time as well, but it's another story about how we achieved that. We could show, in addition to these episodes of progesterone, we could show that they'd ovulated by doing ultrasound scanning, and you can actually see the corpus luteum. That's evidence that they'd ovulated even out of season. And we were able to mate them. So I forgot to say, but the mating that we observed earlier on was with a vasectomized male, so that we wouldn't muck up our experiment, and they would continue to carry on going through their cycles in their non-pregnant state. But at the end of this experiment, we decided, let's see if they can get pregnant out of season, and so we put them with a fertile stag, and this rise in progesterone that just stays up is evidence that they have become pregnant, and we were able to show that with ultrasound scanning. This is an out-of-season conceived red deer fawn, and here it is here. It was born in July, so we called it Jules, and it featured on TV3 News. I think it's the first out-of-season born deer that wasn't born by manipulating the length of day ever. We wanted to So that was all very well, but we wanted to know, are the mechanisms at the level of the brain and the pituitary gland that are driving all these things, are they fundamentally dependent on thyroid hormones as well? And so in a separate group of animals, we measured profiles of luteinizing hormone that I've told you are the driving factors for all of these things. And in our control animals, you can see luteinizing hormone went down and became undetectable in midsummer. This is profound infertility, much more than you would see in a sheep or many other seasonally breeding animals. But the thyroidectomized animals, they maintained high levels of luteinizing hormone right throughout for as long as we could measure. And if you take a window into, if you put a magnifying glass on different times and actually take blood samples every 10 minutes for, well, these are for 9 hours. I have no idea why I chose such a long period of time, but that's an extraordinary amount of blood sampling to do. You could see that in both groups of animals, their LH pulses are going really fast, indicative of fertility during the winter months. And in the control animals, during the summer, LH was completely undetectable, no pulses at all, profound infertility. But in these thyroidectomized animals, if anything, the pulses were bigger and just as fast, and clearly whatever in the brain causes this transition hadn't happened in these animals. It was a remarkable finding, we thought. Well, that was the end of my PhD, and after that I went to do a postdoctoral research with a guy called Bob Goodman in West Virginia. And so I went there with my wife and newborn daughter at that stage, Lydia. And Bob was good enough to allow me to spend half of my time doing the kind of research he wanted to do in his lab, which wasn't too dissimilar to this, but also to spend half of my time following up on pretty much anything that I wanted. And so I was able to carry on answering some of the questions that had arisen during my PhD that I really didn't have either the funding or the tools to address at Lincoln University. So this is West Virginia, is much colder than Canterbury, but there's some indoor facilities there as well for the sheep. It didn't seem to keep them much warmer. So these are what I wanted to do while I was there, was to try and figure out just where in the brain, and I presumed that it was in the brain, that thyroid hormones were acting to bring about these remarkable changes. And so anytime I show you pictures like this, I'll try and indicate it with this cartoon that this is a coronal slice through the brain to show you different parts of the brain. And so while I was in Bob's lab, one of the things I did was to replace thyroid hormones right throughout the hypothalamus, from the front to the back. And we did that by lowering implants down into our region of interest, bilateral, so one each side, and tamped into the end of these implants are a small amount of thyroid hormones that will last there for a month or two. And so in thyroidectomized animals that aren't undergoing any seasonal transitions, we can try and determine what site of the brain it is that thyroid hormones are bringing about these seasonal transitions. And maybe from there, begin the process of learning something about how it is they bring them about. And all of these diagrams, each little pair of dots represents the sites that I put these implants in, actual sites based on sectioning the brains afterwards and finding out where the implants ended up. And all the open dots indicate implants that were completely ineffective in restoring these animals to their normal seasonal transition. So anything with an open dot just remained in the breeding state for as long as the experiment ran for. But these ones here with the closed dots were the sites where the implants were effective in causing their end of the breeding season. And you can see there's two sites. One is here in the preoptic area, close to a region we call the OVLT, and the other site was here in the arcuate nucleus. And anywhere else, even if we were aiming for these sites but slightly misplaced our implants, which you can imagine happens quite often, none of those were effective. Only when we got direct hits in these two sites were the implants effective in bringing out the transition to the end of the animal's breeding season. So we published this without really knowing much about what was significant at those sites, and we published that in 2003. And to my shame perhaps, I only realised just 2 or 3 days ago when I was preparing this talk the significance of this date in relation to these sites, because in exactly the same year, reproductive neuroendocrinology, which turned on its head by 2 studies published in prestigious journals at almost the same time, within a month of each other. And what these publications showed was that in those exact 2 regions of the brain where my thyroid hormones implants were effective, the preoptic area near the OVLT and the arcuate nucleus, lie neurons which produce a chemical peptide called kisspeptin. And it's only produced in these 2 places, and this peptide is the most potent driver of fertility that's ever been discovered. And it holds huge potential for treatment of infertility, and it's being, therapies are being worked up using kisspeptin to drive ovulation, both in animals and humans. But in that exact same year that these were published, I was showing that thyroid hormones are active in these sites of the brain, and the significance of that completely eluded me at the time. It's only recently a colleague of mine in France in 2016 showed that the neurons which make kisspeptin, they have receptors for thyroid hormone. So this is a technique called immunohistochemistry, where you can image the neurons of the brain, these kisspeptin neurons, and also image thyroid hormone receptors. And when you merge them together, you see that they're overlaid. What this means is that these neurons are able to respond to kisspeptin. So presumably, I was delivering kisspeptin to these neurons. They were able to respond to it and bring about the changes in fertility that kisspeptin neurons drive. So, perhaps a missed opportunity. If I'd followed up on my results, perhaps I could have been part of the wave that followed the discovery of kisspeptin, but we can't live our lives over again. So others who've carried on in this field have been able to put all the molecular and cellular steps together, and they now know that the changes in the length of day that happen as springtime approaches, the days lengthen, and that drives the conversion of thyroid hormone from its inactive form to its active form. This is the active form here. And this active form is able to bring about a remodeling of neurons, such as these kisspeptin neurons, and change the gene synthesis of kisspeptin and bring about the non-breeding state. But that was left for others to do, and I came back to New Zealand and moved into different fields. And so I want to move on to my second little vignette now. And this is work that I've conducted here at the University of Otago. It began as a collaboration when I was working for Professor Dave Grattan as part of his lab, but it's become an independent thing which I've carried on throughout my research career since that time. And so this story is about the link between the level of nutrition and infertility. And I think we're all aware, because it's been drummed into us so often, that we live in this century and perhaps the last half of the century before, we live in a time where obesity has become an epidemic. And as well as all sorts of the effects of metabolic syndrome that leads to, it also is highly correlated with infertility, both in males and females, to the extent that if a couple go to a fertility clinic, desperate to conceive, and if their BMI is 32 or above, they probably won't even qualify for funding for that in New Zealand, because the chances of success are so poor. So it's a big issue for fertility. And at the other end of the spectrum as well, with those with an incredibly lean phenotype, such as athletes, people with anorexia nervosa, that body type is also strongly correlated with infertility. And there are many, many couples who are desperate to conceive but can't do so, presumably because of their lack of body fat. And so clearly fat seems to have something to do with it, and you might gather from what I've said so far that you need some fat, but too much is not a good thing. Well, in 1949, a mutation arose in a mouse line at a lab called the Jackson Laboratories in Maine. And so this gene mutation, which no one had any idea what it was for the next several decades, caused the animals to get fat and infertile. So this is the litter mate of that mouse. It's fertile and lean, but this one has inherited the mutation, which no one knew what it was, and it was obese and infertile. And someone at the Jackson Laboratories was interested enough in this to maintain this line. And although it's infertile, you can maintain the line by breeding from carrier animals that don't inherit the infertility, because they've only got one copy of this mutated gene. And it wasn't until nearly 50 years later that someone called Jeffrey Friedman figured out what the gene was that was mutated. But long before that, people did rather crude experiments, and I've been able to do the same thing in my lab, where you simply take some fat from this healthy mouse, and so you need to suck up about 2 cc's of fat, and you give it to this mouse that doesn't look like it needs any more fat, but what it needs is fat that doesn't carry this mutation. And so this fat will grow in the animal, and rather quickly, miraculously it seems, this animal will become fertile, and over a longer period of time, it will become lean. The fertility is restored before the lean body type is restored. And we've done this in our lab, and it's a remarkable demonstration that there is something about the fat of this healthy animal that this animal here is lacking, and that's leading to its infertility. Well, we now know that what that is, that gene that is produced by fat, is called leptin. It's a hormone, and it acts at all of the levels of the control of reproduction that I've talked about. It acts at the ovaries, it acts at the pituitary gland, it acts at the hypothalamus, and it's critical for normal fertility. And so you need to have it, but too much is not a good thing. And so we set out to do some research, firstly, to figure out where is the critical site that leptin acts. Is it here in the brain? And as a neuroendocrinologist, that's where we want it to be, but perhaps it's other places in the ovaries. And so one of the experiments we've done more recently, actually, was with an honours student called Hayden McEwen. And we've just followed a group of animals over a period of months, and every one of these triangles, every one of these lines shows an individual animal, its timeline, and every triangle is where it has a litter. So these are mice, and you can see that mice have a litter just about every 3 weeks, remarkably like clockwork. And so we could follow them over a period of several months. And so these are normal animals on a normal mouse chow diet. And they stayed fertile for as long as we could afford to pay for their upkeep. Well, when we take those similar animals, littermates of the same animals, and we put them on a high-calorie diet, and this has sometimes been called the Western diet because the level of fat and the level of sugar is roughly similar to a Big Mac. When we put them on that diet, they started off exactly the same, but each animal had 2 litters, and then they just stopped. And so this was as they were starting to get obese, and infertility just set in. Well, we suspected that would happen. We were pleased that it did because that was what our basic experimental paradigm had required. But we did something else as well. We wanted to look at some of the signaling pathways within cells that we thought contributed to the effects of too much leptin. And so when you have too much leptin, some signaling molecules called SOX, stands for suppressors of cytokine signaling, kick into gear, and they seem to disrupt all the effects of this hormone leptin. And so we did a study where we knocked out these signaling molecules. We got rid of them to test whether they were the culprit in bringing about this infertility. And you can see in the animals, KO stands for knockout. When we knocked out the gene for that signaling molecule, the animals remained fertile, not forever. In fact, most of the animals became infertile after about 4 litters, but it at least doubled the length of their fertility. And that suggested to us that this signaling molecule SOX is a critical gene for bringing about infertility on a high-fat diet, and perhaps it could be a therapeutic target for overcoming this type of infertility. I suspect that there are other genes as well, and that's why these animals eventually became infertile as well, so maybe it won't be a complete therapy in of itself, but it's a potential target. And so I guess if you don't remember all that, the take-home message is that too much leptin signaling inhibits fertility. You need some, but too much is not a good thing. Well, we've spent well over a decade trying to unravel where it is in the body, the brain or elsewhere, that leptin acts to bring about these effects on reproduction. And I'm very much indebted to a long-term postdoc of mine, although she's gone on to do medicine now. Her name's Dr. Jeanette Quinnell. And she was with me at the outset of these projects and is responsible for the first set of results I'm going to present here. And what she set out to show was that leptin, its critical site of action is in the brain. Perhaps it acts in the ovary, perhaps it acts in the pituitary gland, but the most important site, she wanted to test the idea that that was in the brain. And all of the experiments that I've done since then have relied on some special tricks we can do by mixing and matching different lines of transgenic mice to target whatever receptor or protein we're interested in and knock it out, get rid of it, just in specific cells that we want. So it's very, very carefully targeted removal of genes, and instead of globally knocking them out of all cells in the body. And to do this kind of trick, you need 2 lines of mice. One is a brain-specific Cree mouse, and that just is a way of targeting the knockout that we want to the brain. We only want it to happen in brain neurons. We don't want it to happen in other types of cells in the brain. We don't want it to happen in the pituitary gland. We don't want it to happen in the ovary. So this mouse allows us to target it to the cells we want. And this mouse here allows us to choose what gene we want to knock out. And so we're able to knock out the receptor for this hormone leptin, put them together, and we have an animal that has brain-specific leptin receptor knockout. And the first thing we noticed, didn't happen immediately, was that these animals became obese. And so clearly you need leptin signaling in the brain in order to control food intake. But even before that it became obvious, Jeanette looked at the puberty onset of these animals. And so this graph takes a little while to get your head around, but it's just a timeline. The green line is for our normal control animals. The red line is for animals in which we've got rid of the leptin receptor, specifically in brain cells. And if this line goes up, it means that an increasing percentage of the animals are going through puberty, as measured by the beginning of their reproductive cycles. And so you can see that, as expected, the control animals go through puberty between 35 and 40 days of age, and by, let's say, 42 days of age, 100% of them have gone through puberty, but not one of our brain-specific leptin receptor knockout animals went through puberty. And so clearly, leptin is required to act somewhere in the brain, at this stage we didn't know where, for the onset of reproductive cycles. So this was a very profound finding that we were very excited about. And we thought, well, is that the end to it? But because we're neuroendocrinologists, we're particularly fascinated in this event that drives ovulation, this remarkable LH tsunami, and we wondered whether the change in responsiveness of the brain that causes this could happen at all in these animals. And so we gave them all the estradiol they need to drive this event. So we're doing 3/4 of the work for them, and we just tested whether they could have this huge release of LH. And so I haven't graphed out this LH tsunami because at that stage we couldn't, our assays weren't precise enough to be able to take multiple samples and graph the thing over time. So this is just from a single sample. But you can see our normal controls had a massive release of LH here. This is the average for the whole group. But the animals from which we'd knocked out the leptin receptor just out of brain neurons had no LH tsunami at all. So even this event in the brain, which is critical for ovulation, you can't drive it. Even if you give them all the steroids needed, you can't drive this event in animals that don't have leptin receptors. We were even able to look at what goes on within these GnRH neurons which drive reproduction. And we can't measure the release of GnRH as a hormone very easily, but we can image the neurons. And so these are 3 GnRH neurons from a control animal, 3 from a neuron-specific leptin receptor knockout. The GnRH neurons looked fine, but we're also able to image the level of activity of these neurons. And these little black dots here within the nucleus of the neurons is staining for a protein called CFOS. And when you see that, you know that this neuron is active, and you really only see that at the time of this LH tsunami, because they're active, because they're driving that massive release of hormones. And I want to say that my kids, during their formative years, became very adept at recognizing a busy GnRH neuron. They would come and look over my shoulder at home and say, that one's a busy one, that one's a busy one. So they've become quite good image analysis experts, at least for this specific thing. But all these neuron-specific leptin receptor knockouts, not one of them was active. And so this infertility seems to be happening somewhere within the brain. So that's the take-home message there, is that leptin acts in the brain to control fertility. And there began a rather frustrating decade where we knew that, but that's not good enough for us. The brain is like outer space. It contains a lot of different cell types. And so we tried to knock them off one by one to figure out where it was leptin was acting. And we got a lot of negative results. It doesn't act directly on those GnRH neurons. The GnRH neurons aren't working, but they don't seem to be the ones that are responding to leptin receptors. It doesn't act on those kisspeptin neurons that I said were the most remarkable discovery of this century in reproductive neuroendocrinology. We felt sure that those would be the targets. Nope, not them either. It doesn't act on neurons that make glutamate. It doesn't act on neurons that make this peptide called RFRP. And so you can see a rather frustrating series of negative results coming out from my lab, and we're thankful that we're able to publish them because, you know, scientists are human and negative results don't tend to catch people's attention. And then a master's student began in my lab, and her name was Olivia Egan, and my next hot guess for where these leptin hormones might be acting was a set of neurons down here in a place in the brain called the arcuate nucleus, which is called AGRP neurons, agouti-related peptide neurons. And so we used the same tricks as before to knock leptin receptors out of these AGRP neurons to test whether they might be the critical neurons that leptin uses to communicate its effects to the reproductive system. And we saw a partial effect. So these are our normal control mice. Their reproductive cycles are beginning at about 34 days of age on average. And the animals we knocked out the leptin receptor from, specifically from the AGRP neurons, they did have puberty, so there's fertility there, but they were markedly delayed in their puberty. Okay, you might look at that and say it's only 4 days, but you have to remember that these are animals that have an incredibly synchronous puberty. Humans are most unusual in the wide age of puberty that we experience. And so a 4-day delay is a big deal for a mouse. And that got us interested enough to look at the role of these neurons a bit further. And we started to think, well, there's probably not just one type of neuron that leptin uses to communicate its effects to the reproductive system. Maybe there's several different types of neurons, and maybe just disrupting any one of them is not going to be sufficient to show complete infertility, because leptin will just use a different pathway to get the message there. And so we took a different approach that we were able to do with our tricks with transgenic animals. And we could knock leptin receptors out of every cell in the body, and no surprise there that those animals were completely infertile. They never began their reproductive cycles. The normal control mice began their reproductive cycles at about 34 days of age, just as they always do. But we had an additional group of animals where we were able to make them completely leptin receptor deficient, except for these AGRP neurons. We could rescue leptin receptors just in that small population of neurons here in the arcuate nucleus. And so we're testing a different idea there. We're testing that these cells are all that is required to communicate leptin's effects and thereby information about how much body fat you have to the reproductive centers of the brain. And remarkably, when only those cells had leptin receptors and no other cells in the body did, we were able to restore reproductive cycle onset. It was a little bit delayed compared with the control mice, but nevertheless, every single animal had puberty onset. And so all that is required is this leptin acting on this one population of neurons to convey the effects to the reproductive system. And so I think there is potential for, well, it just so happens that these same neurons are important for control of food intake as well, and so there is potential for developing drugs that not only control food intake, but might restore fertility at the same time, and that remains to be seen whether that becomes a possibility. Okay, for my final little story here, I'd like to talk about the links between stress and infertility. And so it comes as no surprise that infertility is a distressing situation. It's correlated with depression, and it's a silent situation. People don't talk about the fact that they're infertile. But for couples who experience this and who try again and again to conceive, it's hugely distressing. And it's hard to know whether it works the other way. Does the stress itself lead to infertility? Anecdotal evidence would suggest that's the case, but it's hard to really prove it. And probably the best evidence that that is the case is that cognitive therapy approaches that have some success in reducing the level of stress and anxiety and depression have been, in some studies, correlated with an increase in pregnancy rates. But the data is pretty mixed, and as you can imagine, it's so difficult perhaps to really determine that. We all feel we're stressed, don't we? And some of us are fertile and some of us are not. So these things are hard to get good data on. And we wanted to try, using our mouse models, of course, to try and determine whether there was a link whereby stress causes infertility and what that neuronal link might be. So yeah, it's a chicken-egg argument as to whether the stress is causing the infertility. We know, of course, that the infertility is causing the stress, but does it work the other way? So I need to give you a bit of background about these neurons here, which are in a region of the brain called the dorsal medial hypothalamus. And they're called RFRP neurons. It stands for RF-amide-related peptide, but nobody remembers that. And the characterization of what they do has been done by a number of people in my lab. This is Mohammed Rizwan, June Kim, Caroline Ansell, and India Sawyer are some of the people in my lab that have been working on characterizing what these neurons do. And here are some of their data, just very quickly. Firstly, if you treat an animal with RFRP3, which is the peptide chemical that these neurons make, you can increase stress hormones. Corticosterone is the mouse version of cortisol, the stress hormone, and you can cause a big increase in corticosterone just by treating it with the product of these neurons. In collaboration with colleagues in the School of Pharmacy, we made a drug that blocks the effect of RFRP3 with the aim of perhaps developing a therapeutic drug. We called it GJ14, this drug, if you give it together at the same time as RFRP3, it overcomes all these effects, these stress response effects that RFRP3 has caused. What about behavioural type stress responses? So we have, in our mice, we have measures of anxiety, and one of them is simply to put the mice in, we call it a maze, but it's just two boxes side by side. One is darkened. You can imagine mice like that because it's nice and safe and the predator can't get them. And another is an open box with a light shining on it. You've got to be a brave mouse to go out there. And so normal mice spend about 20% of their time exploring the open box. And if you treat them with RFRP3, they spend less than half their time exploring the open box. They're more anxious, presumably, if you interpret this test in this way. And so they like to spend all their time where it's safe. And again, if you treat them with our drug which blocks the effect of RFRP3, you can overcome that and even make them more bold than they were in the normal state. So that's a little bit of background about what these neurons do for stress responses and anxiety. What do they do for reproduction? Well, so this is another graph here of this LH tsunami, which would normally lead to ovulation. And if we give the animals an injection of RFRP3 at the same time, it almost completely blocks that effect. This release of LH would not be enough to drive ovulation. And we have measurements that suggest that in the normal situation, the levels of this thing go down at the time of a normal ovulation, and it's like it's opening the gates, the floodgates for the tsunami, and so normally that's how this huge release of LH occurs. That's one of the ways anyway, is that the levels of RFRP3 go down to permit it, we think. So with that in mind, we set about testing the idea that RFRP3 levels might increase during anxiety, and that might be one of the things that prevents ovulation from happening. So I was just going to say, this has been thought of as the brakes and the accelerator for different aspects of our endocrinology. So it's the brakes for the control of ovulation, but the same chemical might be the accelerator for driving anxiety responses. So this work was done by another honour student in my lab, Sean Stowe, and Sean set about testing the idea that RFRP neurons might be the link which links stress responses to blocking ovulation. And again, he used our transgenic mouse tricks, and so we have our own mouse line where we can localize this thing called Cre recombinase just to the neurons we're interested, and that helps us direct whatever treatments we want just specifically to these neurons and not to any other cells in the body. And this mouse line here allows, when crossed together, it allows these RFRP neurons to become very sensitive to a drug which we can give them, which allows the neurons to be completely removed. So once we cross these animals together and then give this animal an injection of this drug, we can completely get rid of those neurons. It takes about 4 days and then they're gone permanently. And again, the mouse is perfectly fertile and you wouldn't normally notice any difference. But if we do look at the brains, you can see that those RFRP neurons, which are here, blown up here in high magnification, are completely gone in this mouse here that we've made RFRP null. And none of the other neurons around about are affected. So we looked to see what would happen for this LH tsunami in these animals. And so these are our normal animals that don't have any transgenic changes to them. And when they're in their non-stress state, habituated to handling, so completely unstressed, they have this beautiful LH surge and we stop bleeding here at blood sampling at about the peak. If we just give a short period of stress, and all it requires is that we restrain the animals for a short while, during the build-up of this big release of luteinizing hormone, we almost completely block that tsunami of LH. And so this was a sort of an experimental model for stress-induced suppression of reproduction that Sean developed. I know it's not the same as the long-term stress that's the plague of our society, but it's a nice simple model for showing how stress can affect fertility. Well, what happened when we repeated this in the animals that didn't have any RFRP neurons? They can have a perfectly normal surge of LH in the absence of any stress, so they seem to work just fine. But here's the remarkable thing that we discovered. When you expose them to the same brief period of restraint that prevented the surge in normal animals, these animals were able to have a perfectly normal tsunami of LH that would drive ovulation. And that shows to us pretty clearly that these neurons are critical in somehow linking those effects of stress into the outcome of impaired ovulation. And so perhaps these neurons themselves could be targets for overcoming stress-induced infertility. And what remains to be done, of course, is to determine whether some individuals are more prone to this. Perhaps their RFRP3 levels are high at all times, and it doesn't go down at the time of the surge as happens in healthy women, and those studies still need to be conducted. That's just a quantification of all those results. You can see that in the animals with no RFRP neurons, they are able to have a perfectly normal, this is the peak of their LH surge, even when they're exposed to a period of restraint stress. So that's the end of the 3 stories that I have to tell you about my research. I'd like to thank, this is, I think, a complete list of all the students who have studied in my lab. Some are still here now, but many have moved on and gone and done wonderful things in their own right. And I'd just like to, I've mentioned many of them along the way that have been involved in the things that I've told you about. I'd like to make a special mention of my technician, Megan Ingalls, who's up the back somewhere. She's been a long-term technician in my lab, and she's an absolute pillar in terms of making the things happen in the lab when I'm not able to be there, in terms of training up all these new students in the skills that they need. So thanks, Megan. Thanks also to the critical groups that have funded my research, and these are the main players here that have funded my research. These things cost, as you can imagine, tens of thousands of dollars every year, and so we're very thankful for the funding that's been provided. So thank you all for coming and sitting through this, and I hope the science didn't get too over your head, and I just appreciate your involvement. So kia ora tātou, God bless, and thank you very much, all of you, for listening.
Good evening, everyone. My name's Lisa Matasu-Smith. I'm the head of Department of Anatomy, and I'm absolutely thrilled to be here for a number of reasons. As you heard, Greg is the Deputy HOD of Anatomy, and we're a very, very big department. And I have really come to rely on Greg so much for his incredible knowledge of our teaching program, and it's very complicated. And his very cool and calm demeanor when I, as a new head of department, would get stressed out about things. Greg was always like, no, we got it under control. So when he told me that he had the date for his IPL, this was my first opportunity to be part of the academic party and for a member of the anatomy department to give a professorial lecture, and told me the date of it. I thought, uh-oh, looked at my schedule. That's right around the time that I'm going to a big conference in the US. So I looked at the dates and said, oh, okay, it's at the end of the conference. That should be fine. Then when I got the schedule for the conference, ended up realizing that I was one of those lucky people to be scheduled in the very last session of a 5-day conference. So I quickly gone on to the travel agent and said, is there any way that we can change my flight so that I could get out late Sunday afternoon, which was yesterday in the US, and And we were able to do that and get a very quick 1-hour connection in Houston between my flight out of Albuquerque. So I thought, okay, that should be fine. They guaranteed that it was an Air New Zealand flight and that I could make that connection. And then Saturday night, everybody's phones were going off because there were tornadoes in Texas and the South, and flights were being canceled, and everybody was having to reschedule, and I kept checking mine, and it was all okay, it was on time, I was going to be able to get back in time, and got to the airport, and for the first time ever in my life flying United, the flight was going, and we boarded, and we're getting ready to go. Doors closed on time, and I was going to make it. I was texting Greg, I'm going to make it, I'm going to make it. And we started to take down the runway and almost up to full speed, and then the brakes slammed on, and everybody's looking very nervous and thinking, oh no, here we go. We came to a stop. The pilot came on and he said, bit of a problem. There's a coyote on the runway. Only in New Mexico. And as we taxied around, we saw the coyote trot off after a female coyote. So it's springtime, and female fertility and animal models just about stopped me from being here. But we were able to taxi around and take off again. And with a sprint through the Houston airport, I got on the plane and arrived here this morning. So I am very, very pleased to be here to celebrate with Greg. And I think it was very clear to me. The other exciting thing about it is Greg and I are usually talking about teaching or other administrative things in the department. We rarely get to talk about research. So to be able to see your talk tonight and pitched at the perfect level for a biological anthropologist to really fully understand the exciting work that you're doing. And what really impressed me was the involvement of all of the students and the recognition of all the students, which is just another indicator of what an amazing, not only amazing researcher Greg is, but an amazing teacher and mentor to his students. So thank you, Greg. And we have a small memento to mark the occasion.